The present video is related to the stability studies. In this video, we are going to understand regarding the three main terms: stability indicating parameters, stability indicating methods, and the significant changes. Stability studies are performed for the pharmaceuticals to assign the shelf life, to assign the expiry period, and also to demonstrate that the formulation or the pharmaceutical product retained retains its quality throughout the shelf life or expiry period. Stability indicating parameters are required and to be demonstrated for ensuring the drug quality and safety over time. You can take examples of formulations like tablets, capsules, and environmental factors like light, humidity, and temperature. Stability indicating parameters. So we need to have understanding about these parameters. These are the parameters which are critical for the quality. These are also known as critical quality attributes, that is CQAs, monitored during the stability studies. Show how drug quality changes over time under various conditions. And conditions are the changes because of temperature, humidity, and light. Ensures that the drug remains safe, effective, and high quality throughout its shelf life. These stability indicating parameters, or these are the CQAs, or you can say the tests that indicate whether the drug maintains its potency, safety, and quality during storage. So you can list out the CQAs. Then common stability indicating parameters like assay, degradation products, impurities, dissolution, pH, appearance, water content or moisture content, LOD, then friability, hardness, microbial limits, sterility, preservative contents, all these are the stability indicating parameters. Sometimes you may think that appearance, hardness, friability, how these are the critical quality parameters, or how these are the stability indicating parameters. But though these are the physical parameters, these have the impact onto the quality. So the stability indicating parameters are required to be tested during the stabilities. Then stability indicating methods. The method should detect the potency, for example, like assay without the impurities. So HPLC method is there, UV method is there, many methods are there to test the parameters. HPLC, LCMS detects the impurities and degradation products. These are required to be stability indicating. UV IR spectroscopy monitor the chemical stability and degradation. Dissolution testing measures drug release profile over time. pH measurement ensures acid-base stability. Moisture analysis, whether it is water content or loss on drying, monitors water content. Physical test like hardness, friability, appearance, and color evaluation. Microbial testing, sterility and microbial limit tests. Preservative effectiveness testing confirms ongoing antimicrobial activity. The assay method can be called as stability indicating if it is specific for the assay, and it can also detect the degradation. That means if your formulation is having assay of 97% and other things are there in the formulation like water, then impurities, so the method should be indicating those things. The method should be capable enough to detect these changes. Then significant changes. So significant changes means the deviation to meet the quality parameters. Chemical significant changes like change in potency, appearance, degradation products. Then physical color change, precipitation, then change in the hardness, or tablet may become soft, microbiological contamination, sterility failure, loss of the preservative. Then others like pH change, moisture increase, dissolution failure. All these are the significant changes. Like change in assay more than 5% than the initial is the significant change. When there is a significant change, we need to evaluate the stability and we need to confirm whether these significant changes are because of the stability or because of the method. And if there is a significant change, and if because of that significant change the product is not meeting the quality parameters, then we can say that the batch is failed. Significant changes trigger the reevaluation of the shelf life and the storage conditions. Why stability indicating parameters are important? So because these are important to ensure the product quality, detect the degradation, determine the shelf life and storage, to have the compliance with the regulatory authorities for the patient safety and the efficacy. As I have not included the explanation, I suggest you to watch this video slowly, to take some notes from this and understand. I have many videos available on this channel. I have made many videos, so I suggest you to go through those videos. Here I have enlisted all the three things like stability indicating parameters, methods, and the significant changes. So for example, it is assay. Purpose is to ensure the drug strength remains consistent. Methods are HPLC, UV, or by IR. Then significant change will be loss of active ingredient. Then degradation products, dissolution, pH, appearance, moisture content, water content, hardness, friability, microbial limits, sterility, preservative content, effectiveness. These all are the tests for the stability indicating parameters. There are certain methods which are required to be stability indicating. There is required to be a specification, and there should be a mechanism to detect the significant changes. All the stability studies are performed to demonstrate that the product retains its quality and meet the specification throughout the shelf life. I hope this video will give you good understanding about these three terms. Thank you for watching the video.